Hi, my name is Cole Strickling, and this is modeling transport of blood plasma and solutes within the kidney glomerulus. So the objective of this project was to model the glomerulus using COMSOL, and then verify our model using another published model, and then lastly use our model to replicate the effects of diabetic kidney disease. So diabetic kidney disease is one of the most common long-term complications associated with type 1 and type 2 diabetes. And there are a lot of gaps we have with in the understanding of the progression of the damage within the kidney. So here we will take a look at the glomerulus. So this is the glomerulus. The part we are focusing on is this light blue layer, this green layer, and this yellow layer. This is the mesangial cells, this light blue. This green layer is the glomerular basement membrane. And then the yellow layer is the podocytes. So this acts as a filtration barrier. Blood plasma will flow through, and certain solutes will not pass through this filtration barrier, whilst other solutes like glucose, smaller solutes, will pass through. And so the idea here is that what we know from the damage caused by diabetic kidney disease is that with larger concentrations of glucose traveling through, we end up with damage that allows larger solutes out of this area and into the urine. So our hope is to model this and study the transport and flow to hopefully better understand the progression of damage caused by diabetic kidney disease. So here we have our model in COMSOL. You can see we have the three layers: the mesangial cells or mesangium, the glomerular basement membrane, and the podocytes. So blood plasma flows in through the bottom left here, and then filters through and out through the top. So there are three things we needed to focus on here: Darcy's law, transport of dilute species, and our reactions. So the kidney here is consists of fibers and pores. It's a porous medium, and that's where the Darcy's law and transport of dilute species come into play. And then lastly, our reactions. This will be utilized later once we start adding the reactions. And now we'll take a look at our results here. So here we have the pressure gradient. We were able to model this in COMSOL and compare with Hunt's model. The one thing to note in Hunt's model is that they call it the mesangial matrix, and that is just the bottom layer of our model, the mesangium. So that's why we're only seeing flow in this bottom layer here. And we see that we've got a pressure of 35 going over here, decreasing, similar to Hunt's model, 35 decreasing over here to 34.7. Then we modeled albumin traveling through the model, and as we see, we started at 4 grams per milliliter, and as we travel through here, it increases. Ours did go a little higher than 4.04, but we have the same concept here. And then with immunoglobulin A, it increases as we go across up to 2.245 grams per milliliter, which is very close to Hunt's model. So with this verification, we then moved on to glucose. This is a time-dependent study, and we're seeing similar effects with the glucose going across the model. And so our current goals with this project are to continue to with the model and add all the solutes and reactions. Our hope here is to replicate the change in thickness of the layers, and with that change in thickness, we should be able to replicate the effects caused by diabetic kidney disease that eventually lead to larger solutes such as proteins leaking into the urine at detectable rates. Thank you.